In this video, I'm going to show you how to analyze the data obtained with Miltenyi TCR Clonotyping Kit. We're going to start by adding some data files. I have them on the remote server, so I'm going to go to this folder. And here, I'm going to select samples of interest, these ones. There are 40 files, it's 20 samples. I'm going to import them. Rename the dataset. Let's call it TCR beta. And also let's change the pattern for the sample name to skip the common part of the file's names. So only that part will be recognized as the sample ID. That's it here. I'm going to hit create. So now we have our samples added. One more thing we need is to import metadata. And for that, I'm gonna go to metadata, click import metadata, and now I have metadata saved as a file, as a TSV file that I can drag and drop right here. Just wait a few seconds and the metadata will populate our data set. So we see here that matched samples 20, so all of the samples from the metadata has been matched. I'm gonna click import, and now we see all of the columns from the metadata. Now we can just click run, and the files will be uploaded to the project, as you can see here. The next step is to add the dedicated Miltenyi block to the project. So we have the block Miltenyi TCR Clonotyping. I'm going to add it to the project. And here in the settings, I need to select the dataset that I just created. There's no need to change the preset name. And the chains, I'm going to deselect TCR alpha because we only have TCR beta chain, so no need for alpha export. That's it. I can click run and the analysis would start. Let's do it right now. And for the demo purposes, let me just switch to the project where I already have the same data processed. You can see it right here. There's the same samples, but just already done. So when the Miltenyi TCR clonotyping is done, you can open each of the samples, look at the alignment rates, look at the content of the chain usage. You can see here that's just TCR beta. You can go to quality checks, check all of the quality concerned numbers, like the number of aligned reads, the number of reads and clonotypes. You can deep dive into reports if needed, and also look at the QC report table, which is an overall report table for all of your samples. You can see all of the numbers here. And finally, if you want to look at the actual repertoire tables, we have a dedicated block for that. It's called Clonotype Browser. Let's add it to our project. And here, the only thing we need to select is our data set. Now there are two ways of looking at the data. One of them is looking at it by sample, like that. So I can pick the sample here from the list, and I will see the clonotype repertoire for this sample. So you can see I have the number of UMIs and the sequence of CDR3, as well as V, D, and J genes. And I also have an overlap table, which we have right here. And this is the table for all of the samples together combined. So you can see here, for example, the clone number 1 has been found in 5 out of our 20 samples. And we see, again, the V, J genes, and the sequence of the CDR3. That's it, and thank you for watching.